Good afternoon, students. Now, last lecture we have to discuss about the birth control. The birth controls are the two types: temporary method and permanent method. Today, we have to discuss about the infertility. So, infertility means that is the fertilization. So, infertility is defined as the inability to conceive, that is the naturally after regular, that is the unprotected intercourse. So many causes due to, that is the infertility: physically, congenital disease, immunological, or even that is the physiological. These are the effect for the infertility. Physical causes in female: polycystic, that is the ovary syndrome, PCOS, or that is the hormonal imbalance, endometriosis. While these are the different cause due to, that is the infertility. Physical cause in male: less, that is the sperms count, and small size of penis also affect for the infertility. Today, infertility couples have many options to have a child, such as fertility drugs, test tube babies, artificial insemination, IUI, surgical. Different methods are used for in the fertilization. Now, next, IVF, in vitro fertilization. In vitro fertilization, mainly it is a process of fertilization where an egg is combined with sperm outside the body in a test tube or glass plate to form a zygote. Under simulated condition in the laboratory, the zygote are early, that is the embryos with up to 8 blastome years could be then transferred into, that is the fallopian tube for further development. Here it is mainly in outside, it is the fertilization and transfer zygote material is transferred to in the fallopian tube. This method it is called as the IVF, in vitro fertilization. Now next one, ZIFT, zygote intrafallopian transfer. This method also, that is similar for the IVF, is an infertility treatment used when there is a blockage in the fallopian tube, which prevent the fertilization of egg by the sperm. In this method, egg is removed from woman ovary. Fertilization of egg with sperms in brought about outside the body under sterilized conditions to form zygote by the process called in vitro fertilization, IVF. The zygote is then transferred to fallopian tube for further development. So here also it is similar for, that is the IVF. The fertilization blockage of that is mainly in the fallopian tube. That time that is the transfer outside, that is the fertilization of the union of the sperm and eggs outside, that is the and transfer into zygote material transfer to in fallopian tube. This is method, it is called as the ZIFT, zygote intrafallopian transfer. This method. Next method, GIFT, gamete intrafallopian transfer. This method again, transfer of an ovum collected from donor into fallopian tube of another female who can provide suitable environment for it fertilization and development. So your two donor, that is the mainly fallopian tube, the ovum can be collected. This technique called gamete intrafallopian transfer, GIFT, has been developed for the cases in which only the entrance to the oviduct or the upper segment of the oviduct in blocked. In this procedure, ova and sperms are directly ejaculated into the regions of the oviduct, where fertilization produces a blastocyst which enters the uterus via the normal routes. GIFT has a success rate of the about 30% only. So here also that is the transfer for the directed in uterus. Next one, ICSI, intracytoplasmic sperm injection. ICSI is an in vitro fertilization process in which a single sperm cell is injected directly into cytoplasm of an ovum in the laboratory. Here the sperm has to naturally penetrate the egg. So injection can be in the sperm directly in the female body. That time that is the fertilization can be occurs. So this method it is called as the ICSI method. Next, artificial insemination, that is called as the AI, artificial insemination. In this method, some infertility cases, the male partners is unable to inseminate the female due to a very low sperm count. If in the male that is the low that is the sperm count, that time this procedure it is used. This problem can be solved by artificial insemination. In this technique, the sperms are collected from the male and artificially introduced into the cervix of the female for the purpose of achieving a pregnancy through in vivo fertilization inside the body. This is the artificially, that means sperm can be directed there in the male, collected and transferred to in the female body. So because that is the low sperm counting of the male, that time this method is used. Next one, IUI, intrauterine insemination. This method again, in this technique the process is somewhat like that the artificial insemination. The only difference is that the sperms are introduced into the uterine cavity instead of cervix. So directly the uterine that cavity it is inserted, that is the sperms. This method it is called as the IUI, intrauterine insemination. Next, last method, sperm bank or the semen bank. The sperm bank or the semen bank is a place which collects, stored and provides human sperms or semen. The semen is provided by healthy males called sperm donor. The sperms can be collected from the donors and it can be stored. The sperms are stored in sperm bank by cryopreservation method. This method is cryopreservation method. At low temperature, the sperms can be stored. So, last study the adaptation. Adaptation is a legal that is the process by which a couple or a single parent gets legal rights, privileges and responsibility that are associated with the biological child for upbringing of the adopted child. An adoptive parents should be medically fit and financially able to take care of the adopted child. A person wishing to adopt a child must be at least 21-year old, but there is no legal upper age limit for adaptation. So it is also method for the adaptation of the child. So it is complete for this chapter. Now next revision for this chapter. Some questions about the 2 marks and 3 marks as well as the 4 marks will be covered. So just I will revision headings of the chapter and you have to perfect for the main 2 marks, 4 marks as well as the 2 marks, 3 marks and 4 marks will be covered. So you have to open the textbook. So first is the asexual reproduction. In asexual reproduction, 2 methods. One is the gimule formation. The diagram as well as the some few points you can remember. Budding type. Budding also diagrams also important as well as the few points you have to explanation for 2 marks or 3 marks depends upon the question. So diagram also important. Last year or before last year this budding type of the hydra diagram will be asking. Next sexual reproduction. In sexual reproduction human that is a again first one that is the male reproductive system. Diagram as well as the some points we have to explain. It is also 3 marks for the diagram with label diagram that is the 3 marks or 2 marks question and with explanation means that is the 4 marks question it is comes. So next that is the testes, LS of testes diagram as well as the TS of testes or it is the histology of testes like this question describe or sometimes that is the only diagram for the TS of testes. Okay. Accessory ducts. So all these ducts you have to any 1 or 2 ducts direct question will be asking, remember it. External genitalia also scrotum, few points you have to remember. Next female reproductive system, here also diagram female reproductive system as well as the few points you have to explain for the each one for question 4 marks question or 3 marks that the direct diagram only asking, label diagram of the female reproductive system. Okay, few points you have to explain for the female reproductive for 4 marks question. Next TS of ovary diagram also it is important or ovarian histology of mature, and that is the histology of ovary also. Next one, structure of Graafian follicle. So here also diagram it is important, and a few points you have to explain. Diagram it is very important for the 2 marks and or 3 marks question direct diagram only. So different parts you have to remember. Different the question for the 1 marks or the 2 marks. What is meant by ampulla, infundibulum, like these questions, fundus, cervix, like questions will be asking. So all points you have to remember. Last is the memory glands. Here also the functions or the diagram of the memory glands. Next, puberty, sexual maturity in females, males as well as the females. This is also important for the puberty, 1 marks or 2 marks asking. Next important question, that is the menstrual cycle. For 4 marks question it is. Diagram it is not necessary. 4 points you have to explain. Menstrual, that is the phases, 4 phases. Menstrual phases are the 4. First menstrual phase, ovulatory phase, secretory phase, proliferative phase. Like this is the 4 phases you have to explain for the few points. Next, gametogenesis. Here gametes, the first is the spermatogenesis. This diagram also very important, spermatogenesis and a description. Different phases, that is the 3 phases, multiplication phase, growth phase and maturation phase. So diagram it is important as well as explanation. Depends upon the marks you can answer it. Next, structure of sperm. Diagram it is important. Sometimes it is a description also, but mostly diagram it is asking many times. Next, oogenesis. Diagram as well as the explanation it is needed. For female it is the formation of this ovum or production of the ovum, that is the oogenesis. Next, structure of secondary oocyte, unfertilized egg or ovum, the diagram. Next, fertilization, syngamy, that question, as well as the process of fertilization diagram as well as the explanation it is, and mechanism of fertilization also. Few points you have to explain for fertilization of mechanism of fertilization. Next, embryonic development. So here also significance also you can remember, significance of fertilization. Next, embryonic development, cleavage. Okay, cleavage diagram as well as the few points. Blastulation, second, implantation and gastrulation. These you have to remember. Next, pregnancy. So here pregnancy also there are 3 steps. First trimester, second trimester and third trimester. These 3, that is the points for 3 marks question will be asking. Few points you have to explain. Next, placenta and last the parturition, lactation and reproductive health. Different the goals of RCH programs, goals of RCH and can be achieved by the following ways. Next, birth control. Here also is a temporary method. Different types you have to explain. Remember for the first it is the temporary methods. Next is the permanent methods. In permanent method also there are different types. And last, infertility. Here also there is a different use for the in fertilization, IVF, ZIFT, GIFT, long form also it is the asking or it is the explain gamete intrafallopian transfer, or explain GIFT like ICSI or EI or IUI, like this questions will be comes in this chapter. So this chapter it is complete. So next lecture we shall start the new chapter. Okay, thank you.